Good afternoon, all. Welcome to this edition of the Genomic AI Network's Lunch and Learn series in affiliation with the Alan Turing Institute Clinical AI Interest Group for Genomic Medicine. We're very lucky today to have an expert speaker in the form of Dr. Nikolas Pontikos, who's a clinician scientist working at that interaction between genomics, AI, and ophthalmology. He's based at Moorfields Eye Hospital and the UCL Institute of Ophthalmology. And today we're going to hear about his work on developing machine learning tools to diagnose inherited retinal diseases. I'll leave the details to Nicholas. If you do have any questions, please put them in the Q&A section here on Teams and upvote the ones that you want answered the most, and we'll direct them to Nicholas once the lecture part is completed. Over to you. Thank you.
Great. Thank you very much for the kind introduction. So today I'll be talking to you about genetic eye diseases, specifically inherited retinal diseases. So I'm based out of UCL Institute of Ophthalmology and Moorfields Eye Hospital. I must say it's really a privilege to be part of this network, and it's a really great initiative, and I'm very, very happy to be part of this community. So I just wanted to give you a brief introduction to my background, just because probably people on this call don't know me. So I just want to say that I have a background in bioinformatics and computer science, and I've been working now in ophthalmic genetics for around 5 years. I started the EI2Gene project in around 2019, and that's been funded by Moorfields Eye Charity, but more recently an NHS award that just finished that helped develop the product. And now I'm a co-founder of the spin-out from this venture. So today I'll be talking to you about next generation phenotyping broadly, and I thought I would start maybe with an introduction of what next generation phenotyping is. So I think the earliest forms of next generation phenotyping started with facial dysmorphologies, with the projects that people are maybe well aware of, face2gene, Gestalt, and Gestalt match in particular. And these are using AI on images in order to try and predict genes for facial dysmorphologies. This project's been going on since I think it started before 2019 even, and the publication dates from that time. So these are projects which are quite mature in terms of next generation phenotyping. They're really pioneering projects in this space, and I think there's a lot of publications already and people using face2gene or deep Gestalt matcher in the NHS, I believe, for research. So of interest, I think, is that there's been a recent national level pilot in Germany of Gestalt matcher, a 3-year prospective study with 1,500 plus patients recruited, and it really showed that next generation phenotyping is extremely beneficial for improving the diagnostic yield of patients with these very rare facial dysmorphology conditions. Since then, there's been, since the Gestalt matcher, there's been quite a few other next generation phenotyping approaches. Of course, there's Mendelian, which mines primary records to mostly text, I understand, to identify phenotypes of potential rare disease patients. And there's also bone to gene, which is a more recent project, which is for musculoskeletal genetic conditions. And we've recently written a perspective on the different AI approaches for in medical genetics, which you might want to have a look at. So today I'm going to be talking about the application for inherited retinal diseases. So our publication is the proofs of the paper, which I'm currently resubmitted. So it's all going to be online very soon. So you'll be able to read the methodology if you're interested. For now, I can direct you to the preprint, which is online. You can have a look on the website. It's all the information is there. So we're going to be working from retinal scans in order to identify the likely gene responsible for these inherited retinal diseases. So that's the basic idea. First, I wanted to just take a step back and introduce a bit genetic eye diseases to you. So in particular, the retinal diseases. So what we're looking at here are scans of patients affected by retinal diseases. And the different genes here are indicating the different types of diseases. And in particular, you can see that there's different patterns of degeneration. What is important is that there's over 270 of these genes that have been identified so far, so extremely genetically and phenotypically heterogeneous. And in terms of the impact for the patients, they affect vision in different ways, and a lot of them are progressive and lead to complete sight loss. Also, of interest, they are representing around 6% of the rare disease patients in Genomics England. So I just wanted to give you a bit of background about, very briefly, about why we're seeing these different patterns of degeneration. Well, if we go back to the cell types in the retina, depending on which cells are affected by, what genes are expressing which cells, and what cells are what genes are affected, then you can see different patterns of degeneration. Here, for example, showing you one caused by the variants in the BEST1 gene versus the ABCA4 gene, which gives you different phenotypes. So also to contextualize the burden for the UK, we've got around 30,000 patients we estimate are affected. Although the diagnostic yield is quite high compared to other genetic diseases, it's still not as high as we would like it to be. And obviously the cost, because these are lifelong conditions, there's a huge cost and burden. And then the time to diagnosis is still taking a long time, even though we've got very advanced genomic infrastructure now. In fact, we ran a survey for patients to see what the pathway was for most patients, and this was for around 242 patients and carers. And they told us that even to see a specialist is taking them a very long time, roughly 1 to 4 years. Then obviously the commute to go to the specialist centers is also, quite considerable. The multiple visits, changes of diagnosis, and what was important for us is we also asked them how, whether they would be okay and whether they find it acceptable to have AI implemented in this pathway in order to improve it. And of course, we are happy to say that they are broadly supportive of AI. And we just also did a word cloud of some of the responses they gave us about what more information they would like to receive at the point of care. And obviously, they would like to have more information and better understanding about their disease and what kind of treatments or trials are available. So the benefits of timely genetic diagnosis are clear to the people on this call. I think one thing that is important as well is that in ophthalmology, there's a lot of activity around clinical trials and gene therapy in particular, given that the eye is very accessible to gene therapy. So there's a lot of potential there for these patients, obviously, for being recruited to trials and having access to therapies, but also from the pharma side, there's a lot of drive for identifying these patients so that they can be recruited. Genomic testing in inherited retinal diseases has evolved from single gene tests to now whole genome sequencing. And as I said, around 6% of the rare disease patients in Genomics England are rare eye disease patients. In fact, if we compare the genetic eye diseases, ophthalmic disorders, to the other genetic conditions in Genomics England, it is one of the most common, well, not the most common, sorry, it's one of the ones with the highest diagnostic yield. And this is because, in part, of the good phenotyping data that is available, but it's still not, as I was saying, sufficiently high. We would obviously want to do better. We surveyed a lot of experts in the UK about what they thought the main causes of unsolved cases, and as you would expect, a lot of them are reported that variants of uncertain significance are one of the leading causes of unsolved cases. But also, interestingly, they suspect that some cases are getting tested, genetically tested, and that are not actually inherited retinal diseases. Just focusing on the variant interpretation part, everyone on this call is probably familiar with the ACMG guidelines. And one guideline which is often overlooked is the PP4 criterion, which is how well the gene fits, the phenotype fits the gene. And next generation phenotyping really is going to help a lot with this supporting this criteria and giving it a bit more weight, and especially for the genetic eye diseases where the phenotype is really important. It's really because the quality of the phenotype firstly is good, very good, and it has been historically used in this in ophthalmic genetic disorders. This has been used for a very long time as a really important driver of identifying what is the likely gene. So the technology is there for getting very good phenotyping information. A lot of you might have gone to an optician recently and had an optical coherence scan. There's a lot of manufacturers of these devices. This really gives the opportunity for some multimodal imaging. So if we see, and really what's important to realize is that the pathway for inherited retinal diseases is very much driven by imaging. And in fact, imaging is the first thing that is done when patients walk in through the door. So I've just summarized here some of the key sort of stages in the pathway from a clinical perspective. And obviously the first decision is whether or not to do a genetic test. And I'm going to focus in this talk on these 3 points, which are whether or not to do genetic test, whether the gene is known and what is the likely gene based on the imaging, and also if the gene is not known, what is how can we maybe power gene discovery. So I'm going to talk about the applications of next generation phenotyping with ITGene for this. So the objective here is to improve the diagnostic yield. And if we look at the diagnostic yield simplistically as just a number of positive genetic tests over the number of genetic tests performed, then ways of tackling this are, number one, reducing the number of non-genetic patients which are tested, which I mentioned is a reason why a lot of experts think that diagnostic yield is lower than it should be. And so that's tackling the decreasing the denominator. And then in terms of increasing the numerator, then we want to improve variant interpretation pipelines, so reducing the number of VUSs, and also powering the discovery of new genes with matching patients basically that have similar phenotypes to power gene discovery and gene burden approaches. So I first want to tackle the first point, which is the non-genetic patients. And sorry if this is a bit for people who are not into ophthalmology, these conditions may seem a bit esoteric, but these are inflammatory conditions basically, which are often mistaken because they affect young individuals. They are often mistaken for genetic eye diseases. And so what we've developed is a screening tool that, based on imaging, is able to distinguish at an accuracy of 93% whether it's a genetic disease, it's a potential genetic disease. So I'm going to move now to the variant interpretation, which is the core really of what we've been doing with ITGene. And traditionally, you're familiar with the human phenotype ontology. Well, for genetic eye diseases, around 37 of these terms which are specific to the retina. And these are put into the exomizer and variant prioritizers in order to to prioritize likely genes. So this works quite well, and of the successful diagnoses, around 80% are driven by this variant prioritizer. However, the actual process of getting these human phenotype HPO terms into the exomizer is, as you're familiar with, the test order forms. And this is quite a clunky process, which requires some expertise and clinical understanding in order to know which HPO terms are relevant. The other challenge pitfall I want to point out with HPO terms is, in fact, the mapping of the HPO terms to genes, while pretty good, is still not perfect. And actually, we do find that there's a lot of gaps in the knowledge base. And in particular, one point that is been overlooked is that conditions change over time with these genetic IDs, and so HPO terms need to account for this progression as well. So this is what I've just summarized. And what we are advocating for is to complement the HPO term processes with working directly from retinal images using next generation phenotyping. So we've developed an AI algorithm that is using retinal scans, and I mentioned multimodal retinal scans, which means they're different types of images which capture different sort of features of the disease. And we've trained our AI algorithm in order to predict the gene. So I just want to take a step back and talk about the data set. It's a very large data set that is from a specialist center, Moorfields Eye Hospital. And it covers around 63 genes and around 2,400 patients. It's also a data set, and this is something that, again, is showing you how, at the moment, the genomic service works. Patients are coming from all over the UK to be seen at this hospital, as you can see from the map here on the left. And then also, because it's a lot of the population is from London, there's quite a lot of ethnic diversity. I just want to caveat that the ethnic data is not always very complete, but it is still pretty good for a lot of the analysis we've been doing. And that's allowed us basically to use this data set, and we've trained it to this IT-gene algorithm to use these 3 different types of images. The output of the algorithm is a report which gives you the most likely genes, and we suggest to consider the first 5 hits out of the 63 predictions. In fact, we've also validated this in other sites, and we've relied on international collaborators for this with good and consistent results across the board. We've also looked at the interpretability of this, and we've obviously checked with the clinicians for the sanity of these predictions. So we've benchmarked this against HPO, and what we've done is we've gone and we've taken 130 patients, and we've gone and manually annotated them with clinicians, their HPO clinical experts on their HPO terms. And we then did a comparison of what we get from the variant prioritizer using HPO versus the variant prioritizer using just the images, the retinal scans using IT-gene. So this is the grid that a subset, sorry, of the top 20 of the 27 patients, but just to give you an idea that we obtain, where each row is a patient and each column is an HPO term. And what we found is if we look at the rank of the gene, of the correct gene, these are obviously cases where we know the gene. And if we compare the rank of the correct gene with exomiser versus IT-gene, we see that overall IT-gene is outranking the exomiser HPO rank. And in fact, it's doing it significantly better. So we believe this is a novel AI pipeline for that complements well and complements the HPO pipeline. And then for gene discovery, we can also this works well for genes obviously that are known, so of the 63 genes. However, there's 270 genes, as I mentioned, and some of them extremely rare. So we need to take a different approach here, and this is where a clustering approach is used. But what is nice is we can use the IT-gene representation of these images internally, and even though IT-gene has not been trained to identify these genes, it has an internal representation which can be used for clustering, basically. And this is what I'm illustrating here, is a clustering approach where patients with a similar retinal phenotype cluster in the same area. So we've shown that this can work for the genes highlighted here, which have different, which haven't been seen by the gene and are known to the gene, but have specific phenotypes. And what we can also see is that different phenotypes within the same gene can be clustered separately, which could be useful as well for unraveling gene phenotype associations. So I just want to wrap up by saying that we are going to be running a prospective clinical study now, and this is funded by Site Research UK in order to deploy pilot ITGene in clinics. And we want to also trial the potential of getting a point of care prediction, because at the moment, as we know, genetic testing takes a long time to get back to the patient. So this is all done obviously in a safe environment with experts at the moment, but this is something that we would like to look at eventually rolling out to primary care. I'd like to finish by contextualizing a bit how this fits into the broader strategy and picture of the genomic workforce with regards to AI. So even though we've got very cutting edge genomic technologies with Genomics England, we're still seeing inherited retinal diseases, and I'm sure in all the other conditions, that the pathways for patients are still not fully developed. They're still lacking. And so to get access to this service is still not straightforward, right? There's an element of postcode lottery, obviously, where if you live close to a specialist center, then obviously you'll be seen sooner than if you live somewhere more remote. And of course, there's also all kinds of other ethnic and other sort of socioeconomic demographic factors that need to be accounted for. Then the actual process of ordering genetic tests at the moment, which relies on the test order forms, is also not very straightforward, and I would say a bit clunky still, because you need to basically fill in a form, a PDF form, know which HPO terms to put in. Obviously, you have to always firstly copy and over the patient numbers and so on, and then you need to email that in. So all that sort of process is a bit could be streamlined a bit. And especially if we want to integrate next generation phenotyping into this process, we need to think of ways of doing that. And then obviously the interpretation of the results, which relies on MDTs, is still a complex process, which requires a lot of coordination. So I think next generation phenotyping can be of great utility in, as I mentioned in this talk, improving the diagnostic yield, but also helping the workforce when it comes to identifying these types of rare diseases, describing them adequately using AI, and finally, obviously, if this is deployed in integrated in retinal scanners across the UK, which it can be, given that a lot of the retinal scanners now have AI software that can be integrated into them, then we are looking at really scaling the accessibility to this expert level diagnostic service. So thank you very much for listening. I hope I haven't gone too much over time. I'd like to leave you with the conclusions. That we can use the next generation phenotyping and even bypass the need for HPO terms by working directly from medical images. And this really has, I think, a lot of potential, in particular in inherited retinal diseases where the workflow is so centered around imaging and medical imaging. Thank you, and I'll take any questions.
Thanks so much, Nikolas, and audience. Hope you'll join me in giving some digital claps to our guest speaker for an excellent talk. Very interesting and covering loads of parts, especially the translational elements of the challenges associated with trying to deploy something in the real world in clinical AI. Maybe I'll kick off the questions myself. You've talked about the retrospective validation that you did in a multi-country setting, and now you're progressing to a prospective evaluation of how it works in the real world. Now, fresh off, I guess, doing the ethics and research and negotiating with different stakeholders and institutions from there, I'm sure you might agree that one of the themes of clinical AI is the AI of it is the easiest part. That everything else supporting it is the real challenge. What was the most difficult non-technical challenge you've had to try and overcome in trying to get your AI tool into a clinical pathway?
Yeah. So, absolutely. Just to say that we are in the process of doing it. So, even though the app, the i2Gene app, is available on the Heidelberg Retinal Scanner appway, it can be used for research purposes. But however, as you said, getting it into a hospital with all the governance and IT loops that you have to go through, I think is very challenging. I think probably, I'm going to say the same thing as a lot of people would say, which is I think the governance aspects, the paperwork, the processes in place to do this, I think at the moment are different in different hospitals. And if there was a more sort of unified way of doing this, this would be something that would be very useful, I think, at an NHS level.
Right.
I think the IT challenges are also there, because but those are more technical problems that can be solved. It's more about getting the governance and all the approvals in place.
And so you're having to reinvent the wheel every time for a new deployment because there's no
Yeah,
pan NHS agreements for these types of
yeah,
deployments.
exactly. Like a pan NHS agreement would be would be beautiful.
Yeah.
Yeah, if that existed.
Great. Maybe if I'll indulge myself on a second one in that you talked about PP4 in the ACMG
Yeah.
criteria as where this is targeting as things that are pathognomonic in their appearance, perhaps, or highly typical. Um, what I didn't see, or forgive me if I missed it. What strength of evidence is this equivalent to, or what's the strongest strength of evidence that you're able to um, add as a value add to the, I guess the MDT that the clinical scientist or clinician is using to interpret these variants? Are we talking about strong or moderate or supporting?
Yeah. I think we will be talking about, but so again, the question also, as you know, like will depend a lot on the gene, um, but it will be moderate to strong support for the phenotype. I mean, some genes, some phenotypes are so characteristic they really point to one gene. But some, especially the ones which are more phenotypically heterogeneous, I think there we're going to see more. It's going to be more supporting or moderate. So it will be quite. It will be quite. I think gene specific.
Okay, thank you. I've got a pair of questions from Sarah Ennis, one of our friends and colleagues on the Genomic AI Network. Uh, First question, given the noted importance of variants of uncertain significance as a barrier to genomic diagnostic clinical translation, uh, do you see any application of I2Gene to address this issue?
So yeah, so I think this is where the phenotype can tip either way, a variant from uncertain significance to either benign or pathogenic. So I think this is where PP4 is going to be useful, and often that is in the MDTs. This is at least for the inherited retinal diseases, the clinical phenotype sort of expert advice is often will determine whether a variant of an insignificance is either benign for to be benign or not.
Hmm.
So
Yeah.
so actually it's going to we need to run some more some trials and some pilots, but I have a strong feeling that it's going to make it's one of the most well, I know for a fact that it's one of the most important one, and I think the IT gene is going to be able to help a lot in
Yeah.
that. Yeah. Another thing I should mention is also um, the intronic and structural variants, which are hard variants to interpret a lot of the time. Um, that's also something where a gene sort of indication coming from the phenotype can help in having a closer look at the genetics data and maybe we might have missed one of these genetic variants.
Yeah.
So that's another application, potential
Mhm.
application of this. Yeah.
In a related question with respect to yield, when we talk about these AI tools coming into clinical pathways, often the bottom line for whether they get adopted is, can you demonstrate at a service level that there is, an either increased throughput or increased yield that is commensurate with the cost of adoption, whether that be the licensing for the software, the
Yeah.
training and
Yeah.
digital transformation that has to happen around it. Where do you see how good does this need to be for adoption to be worth it for specialist centers for inherited retinal disease, NGS interpretation?
Yeah. Yeah. So with the health economics is something that we've looked a bit at, but we've looked at it more as sort of a pathway, and we in its current status, we think it's already cost effective, but in terms, I didn't include this in the talk, but in terms of, I just want to say, we benchmark this compared to clinical experts, and just the gene prediction for the gene prediction task, which is it's a difficult task, obviously, just to look at a retinal image and to know what gene it is. But
Mm.
we found that the algorithm performed at least twice as better than the genetic experts when it comes to this. So I think this already demonstrates a lot of value there. In terms of the actual cost and price point, I think that's something to be explored. I think a likely per scan or per patient costing may make sense. And in particular, if we are avoiding patients getting sent for genetic testing, which shouldn't get sent, for example, that's something that there can be a lot of cost savings there, or at least advising whether or not patients should get genetic testing. And then I guess the time, if we can reduce the time to diagnosis, for like, let's say if it's taking 5 years, let's say for a patient to 2 to 3 years, I think that's going to also bring, or even less, that's going to bring a lot of value. So I think we need some more evidence, I think, to potentially to demonstrate
Yeah.
this more sort of piloting, and we can do a lot of that on retrospective data through and this is something actually that's quite interesting from a and I'd be interested in the community, if anyone from the community has any thoughts or would like to reach out to me about this, is uh, building this evidence based on, I guess, retrospective data, because it's easier if you can go back to
Right.
hypothetics and say, okay, if this patient had IgG with that retinal scan, that decision would have this decision would have taken place at that point, and potentially, the diagnosis journey might have been shortened, or so
Yeah.
that sort of level of information would be.
In fact, Sarah asks again in the next question exactly on that topic about
Yeah.
where your ground truth is what to what extent might your AI tool be influenced by the winner's curse because you're training the model on well-established genotype phenotype pairings, barn door presentations.
Yeah, exactly. Well, we obviously are looking at we're training it on the genes we know about already, right? So and we need to have to these AI models need exemplar data from each type of gene. So obviously, the approach can only scale up to genes where we have a bit of sufficient data to train it on. If it's novel sort of genes or rare genes, then we need to use a more sort of clustering approach, which I talked about. But I mean, there's definitely there's definitely obviously some bias to what we already know about.
Yeah.
Um,
I mean, is there scope to have, you said most of the pairings we are looking for moderate to strong evidence on that PP4
yeah,
criteria.
yeah.
Would extending that scope to PP4 supporting as weaker evidence still be useful? Or is the ratio of data collected and model training required for that out of scope?
No, I think that's realistic. I think no, I mean, I definitely think that we
Yeah.
can already be useful for PP4. Um, I think in terms of discovering novel genes,
Mm.
I just want to maybe expand a bit on that, because the sort of current way that is in medical genetics of discovering new genes is finding families a lot of the time not in the UK, right, across different countries, which may have a similar phenotype and have a similar gene, or somebody lists, or we list. The other way to go about it is to list the genes, right, that are candidate genes, and try and match that way. The matchmaker exchange and so on. Well, here we can, especially if we've played a bit with federated learning, and what we can do is we can then and federated execution, we can scan databases of patients in different hospitals to identify phenotypically similar patients, which could be used for this for then powering gene discovery. So I think it can be used as a tool for discovery as well.
Mm.
Yeah.
Is there any evidence on the, talking about weaker genotype phenotype relations? Has there any been testing on mosaic forms of inherited retinal disease?
Oh no, we discussed this a bit, but we haven't gone down that path much. We haven't. Yeah, it's not. It's quite. There's some um, dual genetics as well, which we haven't really looked at. Um, but this would be interesting. I think it's going to be extremely rare and potentially challenging to look at, but it's something
Yeah.
we could explore. Um, and if there's anyone on this call interested in that, be happy to discuss.
Uh, our colleague from the Genomic AI Network, Peter Fish of the company
Yeah.
Mendelian, asks, he says, great presentation, and its a technology looks very promising. You mentioned the next step is a prospective study. Have you
Mhm.
considered applying the tech on the scans of more field patients who've had a negative genetic test over the last 6 to 12 months and reanalyzing these? This may be a rapid way of building evidence for your tech, as you'll likely have a decent sized cohort that you could analyze very swiftly.
Yeah, yeah. Thanks for that, Pete. Yes, absolutely. And yeah, what we're doing at the moment. And in fact, it's not fully operational yet, but we want a pipeline so that every single scan basically would get screened by i2gene. Ideally, that would be the ideal scenario. Firstly, by the screening to say whether or not it's genetic, and all this obviously done in a sort of completely ghost, silent observer type, where we're not impacting the patient pathway in any way, but just to see whether what kind of build this evidence base, as you said, Peter, would be very useful. I think another challenge that we talked about is the governance, the IT, but it's also obviously the time. The biggest challenge as well is people getting people to use this as part of their workflow. So making it as easy and as efficient as possible to be used. Because if it's not fully integrated into the workflow or very easy to use, then people are not going to take time to actually use it or even look at the results. So that's something also that finding ways of how to put this into the workflow. I think education sessions is a good way of bringing these AI tools into the workflow. And that's something we're going to probably use it and try and use it in education sessions with research fellows. Because I think a lot of the time the busy consultants are not going to be taking time to use these tools, right? They're going to have to come from other users. So yeah, no, thanks for that, Peter, and great work on Mendelian.
Where is the state of the art in integration of, I guess, genomics education in ophthalmology at the moment? Is this tool going to displace knowledge in terms of you can envisage a situation where if part of the purpose of the tool is to say whether an eye disease is inherited or not to start with, whose task is that taking if there were very broad adoption of this tool, and who's getting cut out of the process, or what efficiencies are being saved on workforce?
Yeah, no, it's an excellent question. So I think there's different ways of looking at it and thinking about it. On one hand, you know, from a patient perspective, I think anything that can improve the sort of pathway and the information they receive, I think is going to be, I think is valued. So I think we need to think about the patient experience first. So if a patient has a choice of whether they are sent to referred, but firstly, the actual decision of whether they should be referred to a specialist center or not is something that can be, done, I think, with AI, or at least signposting to, where what's the most likely pathway. But then the patient should also, I think, have a choice of whether they want to go to a specialist center or whether they want the AI prediction at the point of care. And then, so firstly, I think that's maybe in the primary care, and I don't think that expertise anyway exists at the moment in primary care. So we would be potentially like even enhancing the awareness of genetic eye diseases in primary care with this AI tool. So in that case, I don't think we are. I think with this probably an upskilling there maybe, but we're not displacing this position. What we could be impacting though is obviously the specialists in the specialist centers, because down the line they might be they might be less consultant potentially for a prediction of what gene is likely to be in an MDT or something like this. But I think at the moment there's clearly a shortage of these experts, right? So I don't think there's going to be this is not going to be replacing anyone, at least an expert. In fact, it's going to be as a lot of people working on AI think, is going to be working in a more complementary way as a companion tool. And in fact, the actual specific task of predicting a specific gene from a retinal scan is extremely challenging. So if anything, it's going to, I think, upskill, I think, in that sense as well.
Do you see that there's Eric Topol recently wrote an editorial piece in, I believe, the New York Times, commenting on certain radiology AIs in controlled situations performing better than clinicians, and also performing better solo as a solo AI diagnostician than the clinician using an AI. Is this something that could happen in a pathway that you're envisaging?
Yeah, I mean, potentially, because I guess I just want to be a bit careful about about these whether this, I mean, potentially, I think we would need to have some studies to see whether or not what is the best use of this technology, and whether, I mean, certainly, I think when it comes to variant interpretation, there's no harm in having this as an extra variant prioritizer, right? As an extra gene prioritizer. But if we are going to make decisions, and I think that obviously the earlier you're in the pathway, the more, in my view, the more risky the more risk there is, because if you're moving, but also the more value there is, right? So if you're moving this as close as primary care as possible, and you have a real genetic eye disease patient that is not going to get genetic testing because this AI told it you shouldn't get genetic testing, that's obviously going to be a challenge there. And I think in any case, I think it needs to be rolled out. I think just a long-winded answer, sorry, but I think what you suggested is potentially possible, but it has to be rolled out in a safe way so that this is incrementally tried out, right, and seeing what and I think one thing actually that is maybe lacking in the AI deployment at the moment is more sort of silent deployments of AI,
Like a shadow
where
deployment.
AI, exactly, shadow deployments, where we see what AI is doing, and observing what would be the impact of this AI decision if it was taken into account, and doing ramifications of that, so that it's not directly, so we could, because we want to see it in the real world for a long time, or some time at least, and see what it's doing, observe it before, and understand how it may impact before actually deploying it. I would like to see that more of these kind of sort of silent deployments, because they're not very low risk. They're not affecting, other than obviously the security considerations and privacy considerations. They're not going to be impacting patients. But in my view, that's something I would like to see more of these sort of studies.
Great. Yeah, I think that would get the pipeline down a bit more, from rather than if we can make that the default, that can just be done alongside standard of care, and comparing it makes that evidence generation step a lot more streamlined, doesn't it?
Yeah,
Uh,
absolutely.
right. Well, I don't see any more questions from our audience, so that leaves me with the job to thank you very much, Nikolas, for a wonderfully engaging talk. I loved hearing about the work you're doing and the real world impact that hopefully is just around the corner with the trial you're about to do. I will leave our audience with this link. If you weren't here recently or haven't been on our mailing list, that whilst there are no open tickets just now, you can still join the waiting list for our masterclass of the Genomic AI Network on implementing AI in genomics, which is happening on the Tuesday, the 24th of June. So just over a month away. If you are a NHS employee, there will be masterclasses on AI for dysmorphology from one of your colleagues. In fact, Nikolas, it's Professor Kravitz's team.
Hmm.
Genomics England, Northwest GLH on implementation of the EMED tool in their laboratory pathway, as well as ambient scribe tools, Tortoise and Heidi. I can't guarantee that there will be lots of tickets opening up, but if you join our waiting list, those tickets that do open up will be allocated to on a first come, first serve basis. Thanks everyone so much for joining us for our Lunch and Learn with the Turing AI Institute, as well as the Genomic AI Network. I hope to see you at a future edition of this event. Goodbye.
Thank you.